In this segment, we will discuss that what are the post-histone translational modification which can be helpful in the study of the epigenetic and the DNA structure. Now, as we discussed earlier, as a matter of fact, the histones are important proteins, and histone tails, they can have various fates including acetylation, methylation, phosphorylation, poly-ADP ribosylation, ubiquitination, and the glucosylation. That is, these are the 6 various fates which histone tails can undergo. Now, histone code is, what is histone code? That is the chromatic structure. Thus, gene expression can be modified by means of the histone code. Histone code is the one which can make or modify the gene expression in the cell and the chromatin structure, and they can change the whole story together. Now, acetylation is a process where one of the best studied histone modification is acetylation. Acetylation है क्या? Acetylation is catalyzed by histone acetyl transferases, which is HATs. ये जो हैं, ये क्या करते हैं कि जो एसिटाइल ग्रुप है उसको ट्रांसफर कर देते हैं टू द DNA, briefly known as HATs. MORF, MOZ, MOF, TIP60, and HBO144, HBO1144 using acetyl coenzyme A as a donor group. They use acetyl coenzyme A जो है उसका donor group use करते हैं, and the acetylation process is going on the histone. Now, histone acetylation occurs primarily at lysine residues. Lysine residues जो इसमें हैं उनमें ये change occur होता है, and the histone H4 and HB3 जो residues हैं उनको ये change कर देते हैं. Now, histone acetylation, that is the 2 biological sequences which are present. One is alteration of histone-DNA binding sites. The lysine loses a positive charge in the process. इसमें क्या होता है कि लाइसिन की जो टेल है, लाइसिन का जो रेजिड्यूज है उसमें ऑल्ट्रेशन हो जाती है एंड इट लूजेज ए पॉजिटिव चार्ज. उसके ऊपर जो प्लस चार्ज होता है वो ख़त्म हो जाता है. Alteration of the binding codes of chromatin interacting transcription factors, that is also affected. That is the consequence of histone acetylation. Now, HATs and HDAC, these are the histone acetylation level that depends on the precise balance between the action of HATs and histone acetylases, that is HDAC. That is where one is acetylated and the other one is deacylation. Now, the 4 families of HDACs are there. That is the class 1, HDCA1, 2, 3, and 8. Class 2, HDCA4, 5, 6, 7, 9, and 10. And the class 3, that is the steruline, that is the 1 and 7. And the class 4, that is the HDAC1, C11. So these are the different classes. Now, those of the classes, उसमें क्या है? 1, 2, and 4, they have similar sequences and structure. But the sirtuinins have a different structural homology. जो sirtuin के groups हैं, उनकी homology जो है, उनका जो sequence है, that is different. Now, another process is histone methylation. पहले तो acetylation हो गया ना, अब methylation हो गया. Histone methylation is another well-studied histone modification where methyl group is, as a matter of fact, added to that. And histone methylation is linked to both transcriptional activation and repression. इसमें क्या है कि ये methylation जो है ये ट्रांसक्रिप्शन, एक्टिवेशन और रिप्रेशन दोनों में काम आती है. क्या करता है? एक तो ट्रांसक्रिप्शन भी चलती है और देन एक्टिवेशन ऑफ रिप्रेशन या रिप्रेशन जो है उसमें भी ये काम आती है. Now histone tails, they can become methylated at several lysine and arginine residues. Among the lysine residues, the best studies are K4, 9, 27, 36 and 79 for H3 and K20 for H4. Now histone methyltransferases, that is the HMNTS, they are the histone methylation that is the catalyzed by HMNT ice. And the methyl group can be removed by the group of proteins called histone-DNA, as we discussed earlier, HDMS. So it means that in the process, the HDMS and the HATs, they proceed together and one methylated, another is demethylated, and so a balance is maintained in the cell for the modification of the DNA histones.